Hi, I'm Victoria, a PhD student within Cardiff University School of Medicine's Division of Cancer and Genetics. This masterclass will focus on some of my own published research on whether or not inherited mutations in pattern recognition proteins, which are part of the innate immune system, can impact survival time in bowel cancer patients who are on a very specific form of chemotherapy known as oxaliplatin. I have previously discussed this research in some tutorials I deliver as part of the student selected component module that first year medical students take in small groups. So hopefully this will give you an idea of some of the concept that these medical students are introduced to in the first few years of their studies. It might be useful for part of this session to write down any new terminology that's introduced to you or anything that you find interesting, so hopefully you can explore it later. My research centres around the use of biostatistics and bioinformatics, which is essentially math, statistics and computing that are used to answer biological questions. Therefore, hopefully by the end of this masterclass, you'll appreciate that research comes in many forms outside of the traditional lab-based medical research. Whilst my own interests are focused on all aspects of how patients react to treatment, this particular academic paper was based on the potential role of the immune system in the activity of chemotherapy. This sort of research is a key starting point for the move away from a scattergun approach where everyone is given the same treatment. This is a problem because a lot of cancer treatment is toxic by nature and can have severe side effects, so if we can reduce the amount of people on ineffective drugs, we can hopefully improve the quality of life. So just as a reminder, what is cancer? Well, there is an argument that if cancer was discovered and characterised now, it wouldn't be given the same name. There are some similar characteristics shared between all cancer types. It is essentially a disease that is largely about uncontrolled cell division and cell growth. And they do share the key characteristic of genetic instability, so that's hypermutation, lots of mutations happening quickly and at the same time. These can occur in oncogenes, where genes that were switched off, that are involved in cell division and growth, gain a function or are suddenly expressed more, or the loss of function of tumour suppressors, which are there to find fault within our healthy cells that have gone a bit wrong and essentially cause them to die in a controlled way. We are also seeing the commonalities of increased cell division across all different all cancer types and decreased mortality. So cells are living longer, acquiring lots more mutations and not dying. This is seen across all cancer types, but how each cancer cell behaves is different. Now, commonly you will hear the statistic that's thrown about of 1 in 2 people develop cancer in their lifetime. This is in effect true, but we are seeing more people who are living with and dying with cancer rather than dying from cancer than ever before. Therefore, finding a way to make treatments better for individual patients is important. I focus primarily on bowel, or colorectal, as you'll sometimes hear it called, cancer. It's a disease of the large intestine and is the second most common cause of cancer death in the UK. The most common treatment is a platinum-based chemotherapy known as oxaliplatin. So as you may be aware, the immune system of all vertebrates is separated into 2 sections, the innate and the adaptive immune system. The innate immune system is essentially the first line of defence. It achieves this through physical barriers, inflammation, and the recruitment and activation of the adaptive immune system, and therefore, the innate immune system acts as a non-specific primary response to any non-self proteins it encounters. These include pathogens, such as bacteria or viruses, or the mutated or damaged cells from within the same body. The innate immune system tends to be fast, non-specific, and a short-term response to these pathogens or damaged cells. On the other hand, the adaptive or acquired immune system has a more specific response. This is due to its composition of specialised cells, systems and processes that detect and destroy specific foreign bodies in the human circulatory system. The most well-known and understood component of the adaptive immune system is the creation of antibodies by T lymphocytes. It has been well established that the immune evasion, which is the process by which cancer cells use a variety of different mechanisms to prevent its destruction, generally by the adaptive immune system. A key example of this is the reduction of tumour antigens. These are specific proteins on the outside of a cell that identify it as a tumour that needs to be destroyed, so that less antibodies can attach themselves to those cells and and promote apoptosis, also known as controlled cell death of these cancer cells. The cancer cells may also, through the use of extracellular signalling, so that's signalling outside of the cell, suppress the immune system so it is less effective at detecting cancer cells and other foreign cells in general. The immune system working well has been shown to improve patients' outcomes on chemotherapy. When patients have lower white blood cell counts, which is a component of the immune system, they tend to live shorter lives on these treatments. For the study we're discussing today, we're focusing on a part of the innate immune system known as pattern recognition proteins. Pattern recognition proteins sit on membranes, either on the outside of cells or on the outside of nuclei. These essentially sit on the fatty membranes and act as a cross between the outside and the inside of cells. The job of the pattern recognition proteins are to act as the initiator of processes inside the cell by detecting proteins outside of them. Their primary job is to sense pathogens or detect damage-associated proteins and begin the recruitment of cells and processes required to destroy the foreign bodies that have been detected. They do this through signal transduction, where they start a cascade of processes to send messages through pathways of proteins that are modified based on the binding of the foreign protein to the pattern recognition protein upstream of these pathways. So I've largely said that the adaptive immune system, so the specific, slower part of the immune system, has been shown to have an effect on how long cancer patients on chemotherapy will live. So why did I choose to look at the innate immune system, the faster but non-specific part of the immune system? Well, essentially, there were some interesting looking research studies coming out of some groups in Europe, and a lot of them were actually coming out of France. These studies were showing that 3 pattern recognition proteins, toll-like receptors 3 and 4, and 4-mile peptide receptor 1, which I will call TLR3 and 4, and FPR1 from now on, when mutated in mice, were influencing how long these mice were surviving on cancer treatments. Based on this discovery, these same research groups then sequenced the DNA of some human cancer patients and found a similar pattern. Those with mutations that cause the proteins expressed by the genes TLR3, TLR4, and FPR1, so that these proteins were no longer functional, would die earlier. Now, this was interesting and surprising to myself and my colleagues. So we wanted to see if we could see a similar pattern in our own patients who were UK-based. Because if this is the case, the presence or absence of these mutations may be able to be used by oncologists to predict how useful treatment can be, potentially affecting whether or not a patient would go on a specific treatment based on how long they might survive on it, or even simply give patients a more realistic expectations of what survival time is likely to be on a particular treatment. So, how do we go about seeing if we can see the same relationship between pattern recognition proteins and how long patients survive on treatment in cancer patients based in the UK? Well, as part of my PhD, I have access to the healthy DNA, sometimes known as germline DNA, and tumour DNA sequences from about 2,000 late-stage colorectal cancer patients. These patients were part of 2 medical research council-funded clinical trials, one called COIN and the other COIN-B. COIN and COIN-B were designed to test if continuous therapy, or intermittent chemotherapy, which is given in 12-week blocks with gaps, was more effective, or if adding a new immunotherapy-based drug would further increase survival time. Regardless of how the treatment was delivered, all patients were on a platinum-based chemotherapy called oxaliplatin. The data I have access to comes in 2 forms. The information on the tumour DNA is simply whether or not a patient has a normal, known as wild type, or mutated sequence for a number of genes that are known to affect colorectal cancer patient outcomes. These genes include KRAS, NRAS, and BRAF. The healthy DNA is more extensive. We have sequences in the form of single nucleotide polymorphisms, or SNPs, which are single base changes in known identifiable points across the DNA sequence. These essentially act as a proxy for a much longer sequence due to the fact that the large sections of DNA are inherited from either your mother or your father together. So, once we have this data, we perform a regression analysis which essentially detects the size and direction of influence that an independent variable, in this case the DNA sequence within our 3 pattern recognition proteins, has on our dependent variable, which is the survival time of our patients. This can produce a diagram, such as that on the right, which shows how many patients are alive at any given time within, in this case, a particular treatment group. We can see that these groups are significantly different from each other due to different means, indicated by the dotted lines, and the 95% confidence intervals, which is where we think 95% of the general population's results would lie, which is shown as the shaded sections which do not cross. The fact that they do not cross is an indication that there's a significant difference between these 2 patient groups. So, what did we see when we performed our independent analysis on these patients from COIN and COIN-B? Essentially, the changes in the DNA sequence in our patients' healthy cells for TLR3, TLR4 and FPR1 didn't change how long the patient survived on that chemotherapy. This can be seen in our Kaplan-Meier plot on the left-hand side. See how the confidence intervals all cross over, all those shaded areas blend together? Essentially, this indicates that all of these treatments have similar survival times, which you can also see by the fact that all the lines cross over from each other. So at any point, a similar proportion of patients are still alive. This was supported by work in another group who had a data set like ours, but from another independent clinical trial called SCOT, which was based out of Oxford University. This means we can be fairly confident that our results are correct and not kind of the result of a statistical artifact or some funny maths. We did, however, have some promising positive data. The mutations in the patient's tumour DNA, such as the genes KRAS, NRAS and BRAF, were shown to significantly influence how long patients survive on treatment. Those with KRAS, NRAS and BRAF mutations tended to live less long than those who had the wild-type mutations at the same place. From these results, and the results of our colleagues at Oxford, we can pretty confidently say that it's unlikely to be a true statistical association or mathematical relationship between how long patients survive on chemotherapy and the specific mutations within these 3 pattern recognition proteins. It could be that the relationship is true, but down to a very specific stage of cancer, as the original investigation was a stage 2 trial and ours was stage 4, so our patients were metastatic, whereas the patients in the original trial were largely still one sort of primary tumour. However, by partnering up with these researchers in Oxford, we were able to produce results covering multiple stages of cancer, stages 2, 3 and 4, so when the tumour is together or when the tumour has broken up and metastasised. So this theory is actually highly unlikely. The most likely reason why we were unable to replicate this study is that it might be an artefact from the fact that the previous investigators had too few patients in their analysis. The small sample size has been known to increase the likelihood of false positives in any analysis that is done. This is a good example of when you see sort of small sample sizes in adverts that you need to be very careful. However, the most important takeaway from this investigation is that it's unlikely that the mutations in these 3 pattern recognition proteins will be taken forward to be used in the clinical settings. So how does this relate back to the concept of personalised medicine that I introduced to you earlier on in this masterclass? Well, personalised medicine is the principle that rather than using a one-size-fits-all approach where all patients get the same treatment, we move towards a more targeted approach where we separate or stratify our patients based on different things like age or race or mutation status for certain genes and give them the most appropriate treatment based on these characteristics. We are seeing some instances of this already occurring, for example in colorectal cancer. Some tumour genetics is being used in clinical practice, so mutation status is being included in NICE guidelines for drug delivery. So that's the guidelines for what NHS patients can receive when they have a particular disease. So colorectal cancer patients who are a KRAS mutant, for example, are being excluded from certain immunotherapy drugs. The hope from the investigations that came before ours was that the changes in the DNA of pattern recognition proteins in the healthy cells might be able to be used in the clinic in a similar way. However, because we were unable to find the same results and therefore their results were not able to be replicated, it is unlikely that this idea will have much traction moving forward. This also shows the importance of being able to reproduce or replicate findings, because of this idea of excluding patients who had mutated FPR1, TLR3, and TLR4 from undergoing a treatment of chemotherapy, because they wouldn't live as long, had been adopted by somebody like NICE, patients who could actually benefit from this treatment might have missed out. Thank you for watching. I hope you found this interesting. If you did, I would encourage you to discuss these concepts with your teachers, families, or other students, and do some more of your own independent research.
Hello, my name is Laura Molyneux and I'm a third-year medical student at the University of Cambridge. I'm going to be talking today about what it's like to study medicine, the course structure and content, and what a typical day in the life of a medical student might look like. I'm from North Wales and I went to school at Ysgol Friars in Bangor, where I studied biology, chemistry and maths at A-level. I applied to study medicine at Cambridge because I really liked the course and the city. Cambridge is a bit different to other universities, as when you send your application, you also apply to one of its 31 colleges, and I chose Trinity Hall. So the Cambridge course is 6 years long, with the first 3 years being the pre-clinical years, with teaching in the form of lectures, practical classes and supervisions, and then the final 3 years being the clinical years, with most of the teaching being carried out in a clinical setting. In first year, the topics covered are physiology, anatomy and biochemistry, with short courses in medical ethics and statistics. And then in second year, the modules are neurobiology, pathology and pharmacology, with the courses also in head and neck anatomy and human reproduction. And all of these are assessed by end of year exams. So by the end of second year, you will have covered a broad range of topics and will have learned a lot about how the body works. The aim of these preclinical years is to provide a strong scientific foundation for your future learning. And then in third year, you get the opportunity to specialise in a subject of your choice. So my favourite subject in second year was neurobiology, so I've chosen to major in physiology, development and neuroscience this year, and my minor is surgical and radiological anatomy. However, there's a wide range of options. I've included here some examples of the scientific options, such as biochemistry, plant sciences, zoology and pathology. But there's also a range of non-science subjects that you can choose from. So for example, one of my friends this year has chosen to study bioengineering and another is studying English. And so there's really a wide variety of options and you should choose something that you just find really, really interesting and want to study for the year. And there's also a preparing for patients course, and this occurs in first, second and third year. It's a smaller course and it's your opportunity to meet patients, to practice interviews and also to improve your communication skills ready for clinical years. So in a typical day, I wake up at about 8 AM, I make breakfast and have a coffee, and then I meet the other medics outside Trinity Hall and we all walk to lectures together. We set off at about 10 to 9, so we make it in time for our 9 AM anatomy lecture. So after our morning lecture, we usually have about a 2-hour break, and I quite like going back to college and studying in the Jerwood Library, and that's that big glass building on the river. And since we've just had an anatomy lecture on the embryology of the heart, I'd probably go over my notes for that, make some edits, and focus on any parts that I found confusing. And then at 12 o'clock, we have a biochemistry lecture about DNA repair, so I'd do some pre-reading for that. At 1 o'clock, I come back to Trinity Hall for lunch. And this is a really nice opportunity to just chat with friends and to catch up with people who are doing other subjects because everyone tends to come back to hall for lunch. On the left, that shows our dining hall, and on the right is the garden. And in summer, it's really nice to go to the canteen and get food and then to bring it out and sit on the grass in the sun. And then 2 till 4, we have a physiology practical where we're studying respiration. But this afternoon session could be another practical. It could be biochemistry or histology, or it could be a dissection session. So 4 till 5, I usually go back to my room and I do some studying and I might revise for a supervision that I have that evening. And then 5 till 6, I have a physiology supervision. So the supervisions are the names given to the small group teaching sessions that occur in Cambridge. It's usually about 2 or 3 students and 1 academic, and it's a really good opportunity to consolidate your knowledge on the subject, to ask any questions you might have, and to be tested. And I personally find them really, really helpful. So supervisions occur in college. So straight afterwards, we all walk to hall together and we have dinner and just chat with everybody. And then for the next hour and a half, I'll go back to my room or I'll go to the Jerwood Library and I'll do some studying. I usually have supervision work. So this week I've got an essay on how emphysema affects lung function. So I'll start my research on that and maybe form an essay plan. And then at 8 o'clock, I go climbing. I really enjoy climbing. So about once or twice a week, I organise to go climbing with a group of friends or with the Mountaineering Society at Cambridge. And we'll cycle to the local bouldering centre and go climbing for 2 hours. And then after that, I'll get back and I'll do some pre-reading to prepare for tomorrow's anatomy lecture. And then about 11 o'clock, I'll probably get into bed. I might catch up with some friends from home, call my parents, or maybe read a book and then go to sleep. It's a challenging and interesting and a really engaging course. But it did take me some time to get used to the increased workload. So I'd recommend maybe spending the first term learning how you learn best, whether that be by making flashcards or typing up notes or drawing posters. Just find out what works best for you and try to stay organised. I find having a to-do list and a weekly planner really helps me to keep on top of things and make sure I don't miss any supervision deadlines. But it's definitely possible to find a work-life balance and really important to make time to do the things that you enjoy. And there's a society here for almost everything, ranging from sports clubs and hiking to board games, even a coffee drinking society. So you're guaranteed to find something that interests you and to find like-minded people. I know for sure I've met lots of really lovely people whilst being here. Thank you very much for watching. I wish you the best of luck with your university applications, and I'll hand you back to the host who will let you know about the next steps.
Gobeithio chi fwynhau. I do hope you enjoyed your first masterclass full of interesting ideas. Egwyl fach nawr, let's have a short 5-minute break from the screen. Stretch your legs, take a comfort break, ac yna dod nôl i'r lobby. Come back to the lobby for your next masterclass. A fydd yn dechrau, which will start at 2:55.